Hello, everybody. Thank you for attending today's webinar. INOVIQ is holding the webinar to discuss the outcome of our retrospective exosome ovarian cancer study, what we found, and the specific actions that we are taking before progressing to the next study. The board and management team are all present on today's call, with the exception of Mary Harney, who is currently on flight. We acknowledge the disappointing outcome of the study and are undertaking a comprehensive technical review to strengthen the design and execution of future studies, minimise any impact on development timelines, and support the path to commercialisation and long-term shareholder value creation. The underlying scientific rationale remains strong. A key development risk is being addressed, and the EXO-OC development program remains on a path to optimisation, validation and commercialisation in a partner laboratory. Our commercial strategy is unchanged. INOVIQ remains committed to commercialising the EXO-OC test initially as a laboratory-developed test in the USA for early detection of ovarian cancer, before further developing the test as an in vitro diagnostic for screening ovarian cancer in average-risk women. We plan to address several key shareholder operational and governance questions up front before moving into the presentation. We will then hold a further shareholder Q&A at the end of the session. Let's move on to the next slide. The obligatory disclaimer regarding forward-looking statements. Next slide, please. Slide 4. These questions have been sent in to the company during the last week, so we're going to take you through these key questions. One of the key questions is, why weren't the sample quality issues identified earlier? All samples were sourced against the company's predefined criteria. They were inspected on receipt in accordance with our quality control checks. They were then stored for later sample processing and analysis in randomised batches under a Human Research Ethics Committee approved study protocol. The sample quality issues could not be identified until the sample analysis was complete. The study was then unblinded and initial data analysis was completed on the 27th of June. The unexpected differences in biomarker levels between the biorepository providers was deemed consistent with provider-specific pre-analytical factors. The next question was, what was the test performance in the samples? As the samples were considered unsuitable for meaningful evaluation of the EXO-OC test performance due to inconsistent provider-related microRNA biomarker levels. These differences are most likely attributable to variations in sample collection, handling and storage procedures prior to INOVIQ having received the samples. These pre-analytical factors affect extracellular vesicle and microRNA biomarker integrity and can significantly impact test performance. Another question was, will the EXO-OC test work in the real world? Yes, we are confident that the test will work in the real world. The final EXO-OC test will have a routine pathology biospecimen collection protocol. Every test that is sold through any laboratory has its own collection protocol. It is designed to control sample collection, handling, and storage factors. This will be in place before the test is performed in a clinical setting, in a real-world evaluation, or in a future prospective clinical trial. INOVIQ remains committed to commercialising the EXO-OC test, initially as a laboratory-developed test, followed by an IVD for screening of ovarian cancer. Another key question was, what are our next steps? We are completing a technical review to inform our future validation studies, refine and validate the EXO-OC biomarkers and algorithm, and to optimise the development pathway. We're also progressing discussions with potential partners to support technology transfer, assay optimisation, and final validation of the EXO-OC test in a CLIA certified partner laboratory to enable initial commercialisation as a laboratory developed test. Another question was, what did the study cost? The cost of the 1,255 case control studies was $849,000, and the total project spend was $1.216 million. This resulted in a final cash position of $9.35 million at the 30th of June. No sample acquisition costs were incurred for the previously scheduled high-risk and confounding disease groups. These components will now be in a future study. INOVIQ is actually funded into 2027 and continues to have a disciplined capital focus on advancing the EXO-OC diagnostic and our exosome therapeutic programs. Another question was, what is the impact on our timelines? As I previously stated, we do aim to minimise any impact on our development and commercialisation timelines. It is too early to quantify the precise impact, but our commercial strategy remains unchanged. We are undertaking a technical review and progressing discussions with both contract research organisations and potential partners to support the next phase of development, and we will update the market as these key milestones and timelines are refined. I'll now ask Peter to run through some of the governance questions that we have received.
Thanks, Leanne. And again, I'd like to express on behalf of the board our disappointment. We share the disappointment that all shareholders have with regards to these results. So the first question concerning governance was the board and management oversight of the study. The study was managed under INOVIQ's established product development and governance processes, including HREC-approved study protocols, structured project management and reporting processes, regular risk and progress reviews, and independent data analysis. Upon identification of the study issues, management escalated the findings to the board, which is overseeing the technical view and actions to strengthen future study design, quality controls, and governance processes. The second question, were the development risks adequately identified and managed? INOVIQ identified the key risks associated with retrospective case control studies using biobank samples, including sample quality and pre-analytical factors. Risk mitigation measures were implemented, including supplier qualification, predefined sample requirements and inclusion criteria, temperature-controlled shipment monitoring, sample receipt inspections, and QC checks. Consistent with standard diagnostic development practice, these risks were balanced against the stage of development, cost and timelines to efficiently advance the program to the next milestone. In other words, we made as many preconditions as you would expect us to, but unfortunately, we're unable to confirm that our preconditions were met. Third question, what decision-making processes ensured timely ASX disclosure? The board considers that decisions were made in accordance with the information available at each stage of the program and consistent with the company's governance, risk management, and continuous disclosure obligations. The fourth question, what are the consequences for management? Management is accountable for delivering the company's strategic and operational objectives. This is regularly reviewed as part of performance management. No bonuses were paid in respect of financial year 25 as a result of the poor share price performance. And it's anticipated that a similar bonus outcome will happen for financial year 26 when reviews are completed in October. Other considerations outside of usual reviews on management and board renewal will be made after a full review has been completed of the current study. The last question, we anticipated concerning governance is how will the board restore shareholder value? We acknowledge the recent study outcomes are disappointing. The board believes the underlying scientific rationale remains strong. By addressing the key development risks, identifying, strengthening future study design and execution, progressing commercial partnering opportunities and maintaining disciplined capital management, the company aims to reduce risk, accelerate value creation, and deliver long-term shareholder value. So in summary, disappointing result, but we believe these setbacks are not uncommon, and we believe we can restore shareholder value.
Thanks, Peter. I'm now going to hand over the study review to Professor Greg Rice, who will take you through the actual study and the outcomes. Thanks, Greg.
Yeah, good morning, everybody. As Leon mentioned, I'll take you through the technical side of the project and the data. I'll go through these slides fairly quickly so that we can have some time for discussion at the end of the presentation. Next slide, Emma. Look, I think it's useful first to provide some context on where the EXO-OC test sits in its development. And I've used this diagram from a Roche document that outlines the process of developing a biomarker test. It moves through a very defined pipeline through the discovery phase, through clinical validation, verification, and then on to commercialization. And in our case, that's initially as an LDT. The early studies that we did with UQ and the OC500 study really established the biomarkers and the algorithm. The OC2200 was really the next step in validation towards delivering that LDT in a clear lab. And this is the standard process through which any biomarker test progresses through. And really the arrow there shows us where the EXO-OC assay sits. It's really in a stage of assay refinement and development. Next slide, Emma. So this is really the data that underpins the OC2200 study, why we initiated that study. You've all seen this before. This summarizes the results of the OC500 study, which was a retrospective case control study using samples that were collected under a pathology standard blood collection procedure in Australia. The EXO-OC algorithm was developed and cross-validated using an independent holdout test set, and it was done by one of Australia's leading computational scientists, so an independent scientist. These are the data that were provided by her, and the data that we have presented to you and the market. In developing that algorithm, all safeguards were put in place to avoid overfitting, including regularization. So the computational scientist basically did everything that she could in terms of trying to avoid overfitting. But really the only valid test for overfitting of an algorithm is to validate it in a completely independent sample set. And this was really the OC2200 set itself. Next slide, Emma. So just to remind you of the test, briefly, this is how it works. We use our EXONet immunoaffinity capture beads to capture extracellular vesicles, exosomes, from plasma. We measure 6 microRNA that are associated with those exosomes, and that's measured, the microRNA are measured by RT-qPCR. And we also then separately measure the plasma concentration of CA125. The machine learning algorithm then combines those 7 inputs into a single EXO-OC score, and that returns a binary result. Either cancer is detected or it's not. So that's a simple explanation of the assay. Next slide. In terms of developing a new biomarker test, EXO-OC test, the underlying principles for developing the diagnostic are really not dissimilar from any product development. It goes through a process of design control. And this one that I'm showing here is an FDA ISO framework for design control. And as part of that process, it's initially identifying the user needs. In this case, the clinical need is an accurate blood test for the early detection of ovarian cancer with high specificity. The next step in that process is to establish the design inputs. What are we trying to achieve? And in the design inputs, the criteria that we set there was that the test needed to have a sensitivity of greater than 75% and a specificity of 98%. It's probably worth pointing out here that the FDA has not specified a criteria for ovarian cancer screening. There is no FDA specification around that. The numbers that we use and the numbers that we build into our program really come from prior publication of experts in the area dealing with ovarian cancer screening. So that's where those actual numbers come from. The next step in the process is really building the prototype. So this is design processes, and this is the EXO-OC assay, the CA125 and the 6 microRNA based around that OC500 study. The next step is to check the performance of that test in an independent sample set, the OC2200 set. So we are checking the design outputs against the design inputs. And this process is an iterative process. Emma? Yep. So basically, it's an iterative process that goes from design output to check whether we've actually achieved our design inputs. If we haven't, it's an iterative cycle. And this is a normal part of design control and developing a new product. So the design control provides us with information that we can act on to really improve the product and achieve ultimately a validation of the medical device against user needs. Next slide, Emma. Okay, so this is really the commercial logic, I guess, behind it. It's to really reinforce that design control process that we've been implementing. And this really describes the fastest path to market to generate early revenue and focusing on the LDT. And what the whole process involves is, one, firstly, validating and qualifying those biomarkers, building that assay, so that's the OC500 assay again, analytical verification of that, coming back to this design input process, and clinical validation of that, the OC2200 study. And this is an iterative cycle. If you click on that, yeah, so that's the iterative cycle that we're going through. Once we're satisfied that the design outputs match the design inputs, then we can move on to the next phases in terms of building its clinical utility, providing that evidence, transferring it to an LDT as an LDT test into a clear lab, and progressing down the pathway towards developing it as an IVD. Next slide, Emma. So what happened in the OC2200 test? And this slide actually lays out the whole process on a timeline. And if we move through it, back in October 25, we had planned to acquire samples from a biobank, an academic biobank, in collaboration with one of our academic collaborators. Those samples were to be transferred to us pending analysis of the metadata, the clinical data that's associated with those samples, and that they satisfied our criteria. In December, the sample clinical and QC metadata were made available to us. When we reviewed those, we identified that our criteria for acceptance of samples into the study were not met. In particular, those samples had been stored for greater than 5 years, in fact, more like 20 to 25 years. And the volume that could be provided from those samples was less than the 250 microliters that we need to run the assay. So they neither met the storage criteria nor the sample was available to actually run them. And we made that call in January 2026, which meant then we had to very rapidly identify another source of samples that we could use, and obviously also put in place our own ethics requirements, ethics approval requirements around the project itself. So that process was going on in January. What we ended up having to put in place to acquire the requisite samples was to go to commercial biobanks to deliver those samples. Given the rarity of ovarian cancer, what we discovered was that no single biorepository could provide the samples that we required. So we had to extend the search for those samples over a number of biobanks, and ultimately we had to deal with 5 different biorepositories, international biorepositories, not from Australia, obviously, to acquire the samples. It's also worth noting that those samples didn't come in all at once. They came in over a period of time in multiple batches as we were able to acquire them from the respective biobanks. We actually started isolating the extracellular vesicles, the exosomes, in March. In April, we started measuring the microRNA themselves. The last samples, which was actually a fairly significant set of samples, arrived in May. And the complete sample analysis in terms of having that microRNA readout on all the samples was in mid-June. I then unblinded the study and then completed my initial analysis of the data, which I made available on the 27th of June. That was provided to the CEO, who provided that information to the board, went into a trading halt, and we announced it on the 1st of July. So that really outlines the timeline and what's happened. I think really the important things to note here and to remember is that 5 biobanks, they don't deliver the samples all at once. That's over a period of time. We have to then, 1, extract the EVs, 2, process those and measure the microRNA, and then unblind the study to find out exactly what has happened. Next slide, Emma. Now, one of the issues that we have to deal with when working with biobanks is that we lose the opportunity to monitor the sample collection process. That's done by the biorepository itself, and we're not able to monitor. The samples are already collected when we go to the biobank. And there are a number of things that can happen during that collection process that can impact on the biomarkers that we're measuring, particularly exosomal biomarkers. And that includes the time of blood collection, how long the blood sits around, the type of collection tube that's used, how that's actually then transported from the collection site to the point where it's assayed, how those samples are actually processed, how they're stored, before we get to the point of even analysing them. what we did was provide criteria along that whole pathway to every biobank. This is the type of sample that we want. So we specified what we wanted. What we can't check is anything up to the point of receiving those samples. And it's very different from a prospective study or from an established blood test that's run by a clinical pathologist here in Melbourne, for example. They have complete control over that whole process, and that's how the EXO-OC test would be implemented clinically, and all these we would be able to check and to monitor. Next slide, Emma. So just an example, some of the information we provided to these biobanks, obviously specifying the way that the samples collected, what metadata we needed, the type of tube, how long the sample could be stored for, and the volume requirements. So all of these things were provided to the biobanks. Problem being, we don't have oversight on it. In terms of the patients, again, the patient groups that we wanted were very clearly and prior to the study starting defined, and again provided to the biobanks to inform them what samples we were prepared to accept. Next slide, Emma. Once we've received those samples, we go through our own QC process, obviously. Those samples, obviously, when we receive them, are inspected for their condition, their dry ice status. We run a shipment temperature log so we can make sure that they don't go out of temperature specifications. Obviously, we reconcile against the requirements we provided to the biorepository, look at what they say about the samples. There have been occasions where they have rejected samples on the basis of either there's hemolysis or high lipid content in those samples, and they let us know that they've rejected those samples. They're not in the pack we're receiving. We also do our own visual inspection for hemolysis and lipid content. Once we've received those and we move into the analytical phase where we actually start measuring things, we obviously run a series of quality controls through that process to make sure the EXONet's working fine, the microRNA measurement is working fine, including whole process quality control, where we take a pooled human plasma, make sure it goes into every assay, and we look at how it performs so we can understand the whole process is working perfectly. We also include another step in addition to that to ensure that from the point of extracting the RNA from the extracellular vesicles and measuring the microRNA, that is all consistent, and we use an RNA standard for that. All the tests are run in duplicate to identify aspects of precision. We obviously run everything to our standard SOPs, monitoring how that RNA measurement is behaving, that it's within specification, and then we're able to review the quality of the data itself. Next slide, Emma. Okay, I just want to bring this point home again about the differences between how a test operates in a clinical pathology laboratory, how we will deliver the EXO-OC as an LDT is really shown on the left panel, where there are a number of collection sites. So you can imagine those as the ones down the road where you go to get your blood taken for a PSA test or something. They come back to a central laboratory, and at the central laboratory has its quality management system over that entire system. They're able to monitor how blood samples are collected. They audit that process. They ensure that samples are collected in a consistent way defined by that standard operating procedure. So they have total control over that system. And that's the way we will deliver the LDT. In terms of early development in biomarker tests, standard practice is to go to a biobank. a retrospectively collected reputable biobank and use those samples. That is standard process. And that's outlined here on the right diagram. In the case of the OC 2200 study, we worked with 5 different biobanks. We provided them with exactly what samples we needed, and it is then on them to provide those samples back to us. What those biorepositories are actually biorepository accumulators. They have a whole series of collection sites behind them where they collect samples from, and we have no information. We cannot access information about what sites they are, or that they actually used the protocol that we specifically designed. So in any biomarker repository study, we cannot extend our quality management system over those collection sites as we can when we deliver the LDT. Usually, you're able to confine your sample collection to perhaps one biobank. In this case, where we were looking to collect 2,000 samples for a rare disease, that's not possible. So we have to go to multiple biobanks. We've asked these biobanks to provide us with the SOPs for collection back here and verify it. We have not been able to access that information. Okay, next slide. Okay, so look, what that means is that the samples that we've obtained from those biobanks is variable, and this is really the evidence. On the left panel, what we're showing is the CA-125 concentration in plasma. And when we unblinded the study and started to look at these samples, what we find is that particularly in the ovarian cancer group, those bars on the right of the left panel, the median value that we're seeing in the OC 500, the OC 2200 set is shown in red. In blue, in the case of the OC 500 study, we noticed the median value of CA-125 was somewhat lower. And actually, when I broke that down into the individual providers, what we're seeing is a significant variation that is related to where the samples came from, rather than the fact that they had ovarian cancer. So this was an indicator that there may be some issues with the samples. And on the right panel, this is what I guess was unexpected and surprising, the extent of the variation that we saw with these biorepositories. Again, the results that we obtained from the OC 500 study are shown in blue. The red bars are from 3 biorepositories from the OC 500, and you can see they vary massively, these biobanks. What we've done is to go back and ask questions about, we have data. Our data is microRNA, exosomal microRNA, and how that varies with disease state. What we can do is we can separate out where the variation that we're seeing in those measurements comes from. We can ask questions about, well, is the analysis of microRNA functioning as it should? Yes, it is. We've got quality controls in there, so it's not that part. Is EXONET functioning as it should? And yes, it's working to specifications. Where does that leave us? It leaves us with the upstream part of the whole study, the collection of the samples. And this is why we're saying that, that these samples are compromised in some way. The data we have is that we're seeing lower microRNA signal from these different biobanks. And this is making it unreliable for evaluation of the test itself. Next findings. So that's I guess a high-level summary of the data that we have. This really leads us to the key findings and I guess the recommendations of the study. What we found was that there were provider-specific differences in CA125 measurements, and that they were lower than the measurements we saw for the cancer groups than the OC500 study. The microRNA similarly varied with each biorepository, and quite significant differences between biorepositories. Everything else in the process, from the time we received the samples to the time we measured microRNA, was maintained constant. It was identical. Every sample saw the same process, yet we saw this biorepository variability. That's got to be due to the samples that we're receiving. So this biorepository variations means that it's not possible to evaluate EXO-OC using these types of samples. What are the recommendations coming out of that study? Well, obviously we're doing a complete technical review. We're going through every step, making sure that all the processes are within spec that we have control over. And that will obviously lead to refinement of the biomarkers and the algorithm if it needs. I think the main finding is really the sample sourcing. We cannot rely on multiple biobanks to provide us with consistently collected samples. The solution to that is to return to a single biorepository provider. That may mean reducing the numbers that we apply the test to clinically validate it at this stage. It may mean that we need to go to a prospective study. That's obviously going to take a longer time than a retrospective study. Work with a CRO or a partner around that. And really we're taking advice from our clinical advisors, but also our computational scientists and our commercial partners around that space. We will progress the pre-analytical studies using this approach and then move to updating the analytical and clinical validation studies. To put it in a nutshell, I guess it's summarized here, correct sample collection equals correct test result. So they're essentially high-level key findings and recommendations. I think next slide, Emma. In terms of the next steps, just at a high level, we are continuing with the model that's developed out of the OC500 study. There's no reason for us to believe that that model does not work. It was generated with Australian samples. It was independently analysed. The models were developed independently. We reported those results to the market, as is our obligation. The samples, when they're collected under a better controlled biorepository source in Australia, give us good results. We don't see that massive variation in the endpoint that we're measuring. So we'll continue this. This is specifically, as we've said in the past, developed for early detection screening, and that's the OC500 original model. I think in the company's discussions with gynae-oncologists in the States who are actually dealing with cases of ovarian cancer, in particular high-risk ovarian cancer patients, what they're more interested in is a test that functions similarly over all stages of ovarian cancer. They're dealing with people who've got a 50% chance of developing ovarian cancer during their life. So it's more important to be able to detect those. And the second model that we're working on, and surely I'll speak to this later when it comes to some of those commercial and delivery aspects of the study. What they're interested in is a more broadly sensitive test to focus on high-risk screening. And there are certain commercial advantages around focusing on high-risk screening as another option. Okay, I think that's okay. In terms of how that affects what we said we would do in terms of developing the LDT pathway and the IVD pathway. Really, nothing's changed. The only thing that's changed significantly is to include in the objectives that we are now going to look at both the average risk, so this is the early screening test, but also this high risk group, because it does have some commercial imperative in terms of delivering the test. Really, everything else remains the same in terms of delivering the LDT as an early detection test and its progression to an IVD. Clearly, the timelines are affected, but essentially that's the only change in plans that we have at this point in time. Thanks, Emma.
Thanks, Greg. So I'll talk you through just a couple of commercial slides, just briefly to remind you about the commercial opportunity and why we continue to be excited and committed to developing EXO-OC. So as this slide might be familiar to some of you, as we've previously reported, we're pursuing a dual path commercialisation strategy to maximise speed, access and value for patients. So initially we will be pursuing the laboratory developed test path, where we will be partnering with a laboratory partner, and we're continuing to have those discussions now. We plan to launch initially as an LDT in the US, and that provides us the fastest path to market and early access to patients. In parallel, we'll also be pursuing the longer term, higher value path to IVD that will require pivotal studies and then provide us that broader market reach and clinical adoption. So why are we doing this? It's a significant opportunity. And so if we take the larger, longer term opportunity first, that is the in vitro diagnostic, where we're targeting screening asymptomatic average risk women. So to remind you, this is the same population that currently undergo breast mammography every 1 to 2 years in line with clinical guidelines. So if we look at 9 major markets, approximately 60% of women who are eligible for that screening actually participate. That equates to over 240 million that are getting screened every 1 to 2 years. So that market is huge. So then if we look at the smaller, nearer term market, that of the laboratory developed test, where we will be launching initially targeting states in the US that require clear certification only, we're targeting high risk women. So that's that very high unmet need that Greg mentioned. So those women are likely to have between 1 to 2 tests per annum. And if we're assuming a test price of about $100 US, we're looking at an addressable market of between 2.4 to 4.8 billion per annum, just with an LDT in the US. So as I say, the commercial opportunity is significant, and that's why we're continuing to progress development of the test. So over to Leanne.
Just
You're on mute.
Leanne.
Oops. Just wrapping up, this is a reminder that INOVIQ is not a single product company. We do have a multi-product portfolio of exosome-based diagnostics and therapeutics. This table really shows that our exosome ovarian cancer test is first being developed as a laboratory-developed test for early detection in high-risk women, with the aim to commercialise that with a laboratory partner in the US next year. And then ultimately we'll be developing that test as an in vitro diagnostic for screening in average risk women. That's a longer term process that will require that pivotal clinical trial, and that will take some time. On the therapeutic side, we are developing CAR exosome therapeutics for our most advanced test. Our most advanced therapeutic is for triple negative breast cancer, where we've completed both in vitro and in vivo, so in animal mice studies. And we've also shown proof of concept in non-small cell lung cancer and also in ovarian cancer of our CAR exosome therapy. that's our key focus for the company, is the core exosome technology. We do have other projects, as our shareholders are aware of, our sub-B2M test, which combines with CA15-3, and some other work, but that is the core focus. That is where the company is investing its money in order to move these programs forward. Next slide, please. So in summary, we do remain focused on our high-value exosome pipeline. We are a leader in exosome diagnostics, and we have a proprietary platform technology that underpins those diagnostics and therapeutics. We're focusing on ovarian cancer because it is a critical unmet need, and early detection will ultimately save lives. We're also progressing our high-value therapeutics pipeline, and we continue to focus on partnering our ovarian cancer tests in order to commercialize it in the US. Our priorities remain unchanged. We are basically focused on completing our EXO-OC diagnostic test development and validation in case control studies in the average risk and high risk groups. As we've discussed, it's important that we validate it in the high risk groups because that is the real opportunity where we can get reimbursement for a laboratory developed test, which really determines the success of your test in market. We're progressing our EXO-OC test towards LDT ready status and ultimately US laboratory partnering. And we are also moving our CAR exosome therapeutics program forward, focusing on manufacturing readiness, preclinical data generation in solid tumours. And as everybody is aware, we did appoint a new Chief Scientific Officer early in the year to focus on this area, Dr. Rebecca Lim. We do continue to focus our investment on these priority programs that we believe have the strongest commercial potential for the company. And we are focused on ensuring that we do enhance shareholder value. I'll now pass over to Mark for the Q&A session with shareholders, noting that we've answered a number of shareholder questions up front, both that came through from a general operational and also corporate governance perspective.
Okay, thank you, Leon. I got a whole host of questions, some which have been pre-submitted and some coming through on the forum as well. So I'll get right into it. Given that the 1,200 unique clinical validation samples were sourced across 5 different biobanks, why wasn't a strict pre-analytical risk assessment or a preliminary pilot stability test executed on a small subset, for instance, 5 to 10%, before committing significant shareholder capital and months of operational timeline to the full batch?
Unmute, good. Yeah, look, this is a good question. The pre-analytical issues couldn't have been detected any earlier with the tools available for working with retrospectively collected samples. The samples from the OC 2200 study, we obviously used biobank samples, and they're sourced against predefined criteria from 5 reputable qualified biorepositories. But in choosing these 5 repositories, we're no longer able to monitor that sample collection. I mentioned that kind of earlier. The collection's already done. We tell the provider what samples we will accept, and they provide us samples on that basis. On receipt of every sample, we're able to check the samples as they come in against our criteria. We check that the samples have been transported correctly, and we do that inspection on hemolysis and lipidemia. I think what no receipt stage check can do for any retrospectively collected sample from any biobank is reconstruct that sample's full analytical history at the point of collection, the time between blood draw and processing or handling conditions before we took custody of those samples. The other thing to remember here is that there is no simple bulk measurement that reports on exosomal cargo. Measuring things like total protein, total RNA and plasma do not tell us anything about the quality of exosomal RNA. There is no evidence for that. So we can't run those relatively simple screening tests. Secondly, doing a small pilot on a subset faces the same limitations. We don't have those bulk tests to actually make a call on the statement of the plasma samples that are coming into the assay. And secondly, the variability in the biorepositories doesn't become evident until the study's unblinded. What I mean by that is the samples have to be extracted, the microRNA has to be measured. I'm provided with that data, and then the clinical metadata is joined to that data set. That's the point of unblinding. And as I mentioned in the presentation, that process does not occur until we have all the data, and that was in essentially, I started my analysis in mid-late June. So I think that's addresses aspects of that question. One, you can't do a simple screen on the plasma samples when they come in. There is no evidence that informs anything about microRNA in exosomes. And two, that issue is not identified until you unblind the study and analyse the data. Thanks, Mark.
Okay, thanks, Greg. Next one. At what specific point during the April to June validation window did clinical operations first detect signs of sample degradation or compromised biomarker integrity? And why was the decision made by executive management to proceed with the full-scale testing rather than call an immediate operational halt?
Yeah, I guess I'll draw you back to that slide that outlines the timeline, and I've kind of answered it a bit in that question previously. It's really not until you unblind that study, when you put the clinical data with the actual analytical data, that we're able to start to tease apart where the variability of the data is coming from. That's when we started to tease apart the differences in those biobanks, and then we went back to the OC 500 and pulled out the raw data from there and compared it. And what we saw was that the signal that we're seeing in these samples from these international biobanks is that the RNA is lower. There's less of it there, and then there's massive variability in relation to the biobanks that provided the samples. That's not becoming available until mid-June, when I started to do that analysis. So we went to the board, and the board went to market on the basis of my analysis of those data. The way that we do the analysis, though, is that we do our own in-house analysis, but we then send it out to our independent computational scientist to do the analysis, which she is doing. And that's the information that we make decisions on. So it's not coming from us. It's independent analysis of data that is the final call on the samples. But given what I saw, I thought it was our obligation to one, report that to the board via the CEO, and two, for the board to make a call on that's obviously market sensitive information, and we needed to release it. So that's the process involved in going through that timeline.
Thank you, Greg. Follow on from that, is the technology itself sound?
Yeah, look, certainly I believe it is. From the OC 2200, what we can say is that we've got the biorepository samples, and we use the same batch of ExoNet for the whole study across those 5 biorepository samples. Every step in the process was identical. It was held constant. And despite that, what we see is variation in the microRNA that is directly related to the biorepository. So there's no evidence that the technology is compromised in any way. It's what's going into the technology. As I mentioned, we run quality controls through the whole process from the time we thaw the plasma to the time we measure the microRNA. We run multiple quality controls to assess the technical performance of the EXO-OC test itself. And we find no evidence that it is not working the same way as it worked in the OC 500 study. So technically, yeah, look, I think it is sound. It is giving us the right results. We're amplifying exosome microRNA efficiently. We're able to measure it with precision at the end, but it's what you put into the assay that determines what you get out. What it doesn't tell us is, have we clinically validated the EXO-OC 500 algorithm? No, we haven't, because the samples in this study weren't fit to do that. And this is really the next step that faces us. We'll do that whole technical review, but the next step is really to obtain samples that were collected in a similar fashion as the EXO 500, the OC 500 test, which were collected by University of Queensland from reputable hospital-based sites, actually, under strict SOPs. And we have those SOPs. We know how they were collected. It's that type of routine pathology-grade collection that we will test on in the next study.
Okay, thanks. Can we still trust the earlier OC 500 results, or was that data set unusually favourable and possibly overfitted?
Sure. Look, we've got no reason not to trust it. As I mentioned, those samples were collected in Australia under standard operating procedures. The results were analysed and the model developed by an independent computational scientist, and they were strong performance, and we reported that to the market as we are required to do so. The OC 500 algorithm was developed under standard acceptable machine learning practice, that is to guard against overfitting. It's something that we've discussed with the computational scientist. It's one of the important things about an algorithm, that it is generalizable, that it works on population or samples from different populations. So that was certainly a criteria that was factored into the design of the model. We went through industry standard validation of the model, that is developing the model with a holdout test set. Once you've developed and finalized, locked the model, you apply it to the independent test set. And that's the data that we show. That's on the test set data. So there is no real reason for us to believe that technology is not working and giving us the results. I'll come back to that point I made at the end of the presentation. Samples correctly give us the correct assay result. I think the short answer to that is we have no reason to doubt the OC 500 results. And two, yes, every precaution was taken to limit overfitting. But again, the final validation of that algorithm is on an independent clinical sample set.
Okay, thanks, Greg. The company previously stated that for an early detection liquid biopsy test to be clinically and commercially viable, it must maintain a sensitivity greater than 75% and a specificity of at least 99.6%. With the recent algorithm tweaks reducing specificity to 98% on the historical 500 sample cohort, how does management intend to market this to test to US clinicians, given the significant higher rate of false positives?
Yeah, I'll take that. As Greg has actually already explained, the criteria that we used, or performance criteria that we used for developing the test, which was greater than 75% sensitivity in detecting the disease, and greater than 99.6% specificity in correctly getting the healthy controls and benign disease correct. That actually came from academic publications and expert reviews on ovarian cancer screening potential performance statistics. Currently, there isn't any performance criteria for ovarian cancer screening that has been set by the FDA or by the US Medicare for coverage of those tests. The reason being that there isn't any ovarian cancer screening test that's currently available and approved by the FDA for screening ovarian cancer. Because we're initially developing this test as a laboratory-developed test for early detection of ovarian cancer, part of our commercialisation strategy and our various partnering discussions that we've had is that we need to be able to provide gynaeco-oncologists with a test that has appropriate sensitivity across all stages of ovarian cancer. Effectively, gynaeco-oncologists, particularly, they're seeing a lot of high-risk patients. So they're women that have got BRCA1/2 mutations, for example, also Lynch syndrome or a family history of ovarian cancer. Overall, they represent around 5% of the general population. In these women, the risk of ovarian cancer can massively increase to as much as over 50% lifetime risk of developing ovarian cancer. What it means is because the prevalence then of ovarian cancer is much higher in this high-risk group, we don't need to achieve 99.6% specificity. Hence, we dropped the specificity, which down to 98%, in order to get improved sensitivity across all stages of ovarian cancer. So it's a specific strategy in order to develop the test for ovarian cancer early detection, particularly in high-risk women where we can detect all stages. What is important is that, as I've said, Medicare in the US will not reimburse an average risk screening test when it's an LDT. That will only happen if a test is developed and approved by the FDA as an average risk screening test. Therefore, as an LDT, we need to be able to have data and a claim that supports Medicare reimbursement. That essentially is, there's two areas that they'll support reimbursement. One is in high-risk groups and the other is in symptomatic testing. So the test is still being developed for early detection. It's just we need to validate the test also in the high-risk group such that we can actually get reimbursement. And there's tests available on the market already for high-risk screening that are laboratory-developed tests that are achieving over $1,000 per test. And that is the market that we're originally screening for. And as Emma pointed out, that's effectively somewhere between a $2.4 and $4.8 billion opportunity if the test was at $1,000 per test. Thanks, Mike.
Thank you. Moving to a US LDT model is an operational pivot away from the original broad regulatory pathway. What are the specific minimum performance criteria required by your prospective US CLIA Labs certified laboratory partners before they will sign a binding commercialization agreement?
Yeah, I think firstly, it's not a move away from our commercialization strategy. The company has continuously informed the market that we do have this dual path commercialization strategy. First, commercialize EXO-OC as a laboratory developed test in the US for early detection of ovarian cancer in high-risk women, and then to further develop the test as an in vitro diagnostic for ovarian cancer screening in average risk women. I think I just explained that because the prevalence of ovarian cancer is higher in that high-risk group, you don't need the same level of specificity, but we need to increase sensitivity across all stages. Again, there's no set criteria. That is something that we are developing with our clinical experts and our partners in order to provide the test really to meet clinicians' requirements for a highly accurate early detection test in that high-risk group.
Thanks, Leanne. Another one. What is the status of your partnering negotiations and will the study outcome impact partnering?
I can take that one regarding partnering. Thanks, Mark. So look, the study outcome really doesn't change or impact our partnering strategy. It might somewhat delay the signing of a definitive commercialisation agreement, but it really doesn't disrupt our plans or the process overall. So throughout this year, as we've informed the market, we've been meeting with a number of different CLIA labs in the US and really assessing them as potential prospective partners for us to bring the test to market as an LDT in the US. So really, those discussions are still ongoing, and we remain committed to developing this test and still very excited by the commercial opportunity, as our prospective partners are as well.
Thanks, Emma. You shifted from asymptomatic screening to high-risk women from IVD to LDT. Does this mean you don't have confidence in the test's ability to be used for screening/IVD?
No, I think as we've explained, we've got high confidence in both pathways, and we're pursuing both in parallel. So as we've always communicated, we are pursuing that dual path, initially commercialising EXO-OC as an LDT for high-risk women. That's the fastest path to market. It's going to ensure early adoption and hopefully reimbursement, while we then undertake pivotal studies, use that real-world data from the LDT to then form a package that we can submit to the FDA for the IVD for high-risk screening.
Okay, thank you. And will this now set you too far behind competitors?
As you can see, the size of the commercial opportunity is really huge, so it really doesn't change the outcome or our partnering strategy or the way that we're pursuing the LDT as that initial faster path to market. So no.
All right, thank you. A question here: how will we restore shareholder value?
I'll take that. So again, whilst the study was disappointing, the underlying scientific rationale remains strong. We are addressing the key development risks that we have identified. We intend to further strengthen our future study designs and execution of those studies, as well as to continue to progress our commercial partnering opportunities. And ultimately, the company aims to reduce risk, accelerate the test towards these milestones, and ultimately to deliver value. We believe that this opportunity is very large, as we have pointed out. The test works. We just need to continue to clinically validate the test in the appropriate samples, which Greg has taken everybody through, and we will design studies appropriately with our advisors in order to do that in the next study.
And if I can add to that, Liana, sessions like this are how we restore shareholder confidence and therefore shareholder value, by being completely open and keeping the market fully informed.
Okay, thanks, Peter. We've got some general questions coming in around CEO and board shareholdings, and I'm probably best to answer those. You would have noticed recent 3Y notices have confirmed significant buying activity across the board by directors. Furthermore, I can confirm both management and the board have participated in capital raises, whether that be placements or SPP in recent raises. And to address the CEO specifically, Leanne participated in last year's SPP, acquiring 171,000 shares, and that was in addition to purchasing 150,000 on market in FY25, which was also confirmed in last year's annual report. Go to some more questions coming through. Given that INOVIQ new pre-analytical factors are a major risk in exosome microRNA studies and had QC procedures in place, why were more rigorous standardized provider agnostic quality controls and real-time biomarker integrity checks not implemented earlier in the sample sourcing, receipt, storage process?
I think we dealt with that one, Mark.
Yeah, okay then. What is the update on the confounding study?
Oh, okay. So that's the confounding disease study. Yeah, look, I think Leanne mentioned right up front that We do plan to do those, but we've stopped any acquisition of those samples until we go through the full technical review. We do that further clinical validation on the OC500 model using appropriately collected samples.
Okay, thanks, Greg. Okay, what incentive does the board and management have to perform while the salaries are much higher than ordinary working Australians? And why aren't incentives more tied to revenue generation and the success of the company?
Perhaps I can deal with that. As we previously stated, in financial year 25, there were no bonuses paid because of the share price performance, and the chances of there being bonuses this year are probably fairly low as a consequence of this recent outcome. And to add to that, we've got to be competitive with attracting the right staff, and if you pay peanuts, you get monkeys. So I think everyone who's got any commercial bent understands that.
Okay, thank you, Peter. Another question coming through here, which is probably best asked or answered by me. It's asking about over $2 million was spent during FY26 in cash outflows relating to admin and corporate expenses. Can you provide clarity on the breakup of this cost? It's 1.2E in the appendix released yesterday. The lease cost for the site can't be that high on Normanby Road. So what is the line item? Okay, so that 2.177 million is a whole host of various expenses. It includes our patent and IP fees, our insurance costs, various professional fees ranging from legal and audit fees. We've got what else in there? We've got computer share registry fees. We've got ASX and ASIC costs. There's IT costs and some other general consulting costs in there, even where recruitment's used, it's in there, director fees as well. So it's really a large bucket of what a typical corporate and overhead costs for a listed entity. And I think those categories would cover well and truly 70 to 80% of that 2.177 million. And in terms of the lease cost, that's a separate line item directly under that. That was around 179,000 for the year. So yeah, that's captured in there as well. And the next question is probably best answered by me as well. It's will there be any compensation from the 5 biorepositories for providing samples which companies state does not meet the predefined criteria and cost 850,000? Look, INOVIQ provided predefined sample collection requirements and protocol inclusion exclusion criteria. The biorepositories delivered samples that met those criteria. However, differences in collection, processing, and other pre-analytical factors, which Greg ran through before, can occur between providers and are outside the control of INOVIQ in this retrospective study setting. So on that basis, we won't be seeking compensation from the providers for the samples, as the samples were inspected by INOVIQ for damage and visual hemolysis and lipemia upon receipt. Our focus will be on continuing development of the EXO-OC test, and this includes planning and executing the next studies to further optimize and validate the EXO-OC test. Question here, when do we expect to see a full review on management positions complete?
I think we've answered the question previously that INOVIQ will update the market as we progress this technical review, and we will keep the market informed of what our upcoming milestones, costs, and timelines are expected to be.
Okay, thanks. The company no longer shows sub-B2M CA15-3 program in the pipeline. Is this program now shelved, and how much was spent on this program?
I'll answer that in terms of where the program's at. I think we formally said in the previous quarterly, in actual fact, that we were still undertaking an optimization of that test, basically using an immunoassay type method that we can use from a, let's say that's more commercially viable. That process is ongoing. We have reduced expenditure around that program whilst we focus on the EXO-OC development and validation, but the program is ongoing, but it is not one of our core assets as we have described.
Yeah, and in terms of expenditure on that program for the financial year, it would have been, look, circa 200,000, so not as significant as the EXO-OC program. Based on the management of sample procurement, it appears management doesn't have enough skill to manage the study. Why not bring in people who know what they're doing and can actually deliver the studies moving forward?
I'm going to respond to that. And I don't think we should get into ad hominem types of discussions. We've answered the questions as to why this disappointment occurred, and we've answered it, in my opinion, in accordance with how any management team would have dealt with this process. So no more questions around that as far as I'm concerned. Thanks, Mark.
Okay. The comments are discussing with potential partners. Who are the team actually talking to, and how interested are they really in supporting future commercialisation?
So I'll handle the partnering discussion. So look, obviously I can't disclose who we're speaking to yet, but we will update the market when we've got, a definitive agreement signed. We are having, as I've said, multiple discussions with US CLIA lab partners, and those partnering discussions are ongoing.
I think just adding to that, the focus basically, in order to progress this test as for LDT commercialisation, once we've completed our own internal clinical validation, we have done further studies around the pre-analytical conditions. The partner that we are speaking with ultimately we have to go through a process, which is technology transfer into that lab. They'll then optimize the test within their own laboratory equipment and procedures, and then validate the test before commercializing it as a laboratory developed test for early detection of in high-risk women. So that's the focus of what all of our discussions are with potential partners, and we do have a number of interested parties. They remain interested, and we will work through any changes in terms of that technology transfer that are required, the pre-analytical studies that we plan to do, whether they are done with the partner or whether they're done separately with a CRO before technology transfer.
Thank you. Is the OC 500 trial able to get above 99.6% specificity? Is there a next iteration of the assessment?
Yeah, look, on my analysis, it doesn't achieve our specifications that we set on the input design. We've explained our understanding of the cause of that. That is what we put into the assay, and we're addressing that.
Thanks, Greg. Why can't the test be provided twice, once for minimal false positives and one for increased sensitivity across all stages?
Yeah, you want to repeat that one, Mark?
Yeah, sure. Why can't the test be provided twice, once for minimal false positives and once for increased sensitivity across all stages?
I don't know, but maybe Emma and Leanne can comment on this, but I know of no test that's conducted that way and commercially viable.
I think the reality is you develop a set of performance requirements, which Greg discussed at the very beginning, which is our design inputs. And the design inputs set what is the minimum sensitivity across all stages. We also have a design input for minimum sensitivity that we wish to achieve in stage 1 and 2, which is early stage ovarian cancer. And then we set a minimum specificity, which is to accurately identify women that are healthy or have benign disease separately to those that do have disease. They're our design inputs. They do come from various sources, and as we've already discussed, partly they've come out of academic publications and also expert reviews of what's required for a clinically acceptable ovarian cancer screening test. To date, there are no clear FDA guidelines around ovarian cancer screening. There are other tests in development, and I'm sure the FDA will move forward to provide some guidelines at some stage around its expectations for tests. If we look, let's say, at other screening tests, so colorectal cancer screening, we are aware that for colorectal cancer screening, that there are very clear guidelines around what is acceptable, and particularly from US Medicare in order to get reimbursement. And effectively, a test has to achieve 80% sensitivity for colorectal cancer screening and 90% specificity. So that's what happens when you actually have tests on the market. Then specifications become clear. When you are developing a new test that doesn't have specifications, they have to really be developed in collaboration with clinicians and with your potential partners, which is what we're doing.
Thanks, Leanne. A question here. Did the test work on the bad samples?
Sorry?
Did the test work on the bad samples?
Well, look, what I've said is that those samples are not fit for performing the test on. And essentially what they report is biorepository variation. In terms of, did we amplify exosomal microRNA out of those samples? Yes, we did. But what they don't tell us really, because of that biomarker variation, is the disease story. Again, I come back to the point I've made before. You've got to collect the sample correctly to get the correct result.
Okay, thank you. What was the rationale for arranging the announcement of the study outcome to specifically not take place last financial year, but rather on the day of the new financial year by using a trading halt?
I can answer that. That was purely coincidental. We were obliged as a board and as a company to keep the market informed, which is those dates are purely coincidental.
Okay, thank you. Dates, timelines are missing from the slides. Why is that? Could we have at least not provided, general targets like H1, H2?
As we've said, given that this information is new, we're still conducting our technical review. We will update the market when we've completed the review and the impact on timelines.
I think most of the salient questions have been asked and answered. Unless there's a genuinely new category, I'd be suggesting we could wrap this up fairly soon.
Yeah, look, I think that there is a few unanswered, but they are largely related around to the sample acquisition, which we obviously cover the reasons and the detail behind the circumstances. So,
Good. Well, in conclusion, I'd like to thank all those shareholders for attending, and we do hope that we've answered the main questions as to why this test was disappointing, or why these results were disappointing, I should say. And we will continue to keep you fully informed as we work towards restoring confidence in shareholder value. Thank you.
thank you, everyone.